Good afternoon, sir. Good afternoon, classmate. We are present by reporting about DNA genetic engineering. Genetic engineering, also called recombinant DNA technology, involves the group of techniques used to cut up and join together genetic materials, especially DNA, from different biological species and to introduce the resulting hybrid DNA into an organism in order from new combination of heritable genetic materials. Genetic engineering is used for many different purposes in research, medicine, agriculture, and industry. The technology is important because it enables the creation of multiple copies of genes and the insertion of foreign genes into other organisms to give them new traits such as antibiotic resistance or a new color. Technology involves the group of techniques used to cut up, join together genetic material, especially DNA from different biological species, and to introduce the resulting hybrid DNA into an organism in order to form new combination of heritable genetic materials. Steps of recombinant DNA technology. DNA isolated in its form, from which means they are developed of together macromolecules. Cutting of DNA restriction enemies degradation. Amplifying of DNA during DNA. Insertion of DNA into house recombinant cells isolation. Informoran and human growth hormone. It is also used to produce clotting factors for thickening hemophilia and in the development of gene therapy. Recommended DNA technology involves using enzymes and virus laboratory techniques to manipulate and isolate DNA segments of interest. This method can be used to combine or splice DNA from different species or to create genes with new function. The resulting copies are often referred to as recombinant. Recombinant DNA is used to identify, map, and sequence genes and to determine their function. DNA probes are employed in analyzing gene expression with individual cells and throughout the tissue of whole organism. Recommended DNA technology is the joining together of DNA molecules from two different species. The recombinant DNA molecule is inserted into host organism to produce new genetic combinations that are the value of science medicine. For example, insulin is regulating produced by means from recombinant DNA with bacteria. Human isolate gene introduced into a plasmid which is introduced to bacterial cells. The bacteria will then use the circular machinery to produce the protein isolin, which can be the distribution to patient.